Assalamu alaikum, everyone. I'm Haleema Shafiq, a third-year medical student, and I'll be helping you with the topic of urea cycle today. So an overview of urea cycle is that you ingest proteins, right, which are the building blocks of almost everything in your body. And those are broken down into amino acids. Now, when you have those amino acids, they are then catabolized to form ammonia. Ammonia actually is toxic to your body, which means you can't allow it to accumulate inside your body for a long time. So in order to eliminate or excrete it from your body, it has to be converted into a less toxic substance, which is urea. So the urea cycle essentially talks about how we take ammonia, which we get from the catabolization of amino acids, convert it into urea, and then eliminate it from the kidneys. So this gives us an overview. We have ammonia, which goes to the liver and then is converted into urea, and this urea is then eliminated from the body by the kidneys. Okay? Now, if we were to go into a bit more detail about the urea cycle, then this is what we see. We'll get to this in just a few minutes, okay? But first, we have to talk about how ammonia gets to the liver and how it gets to the liver, and then it will be converted into urea. But the first step is how it gets to the liver. So we said we have amino acids, okay, which can be broken down in the muscles or somewhere else in your body. When you break down amino acids, you get ammonia, which we mentioned is toxic. So you can't allow this toxic substance to just enter the bloodstream, right? What you do is you combine it with another substance to form a non-toxic carrier, which can then travel in the bloodstream and then reach the liver. So that substance, which we combine the ammonia with, is glutamate. So glutamate is the substance we combine ammonia with, and we form a non-toxic carrier, which is glutamine. Glutamate plus amino acids gives us, glutamate plus ammonia gives us glutamine, and this reaction is catalyzed by glutamine synthetase. So we can see here that you have a muscle, right? And then you have ammonia here, which came from the breakdown of an amino acid. It combines with glutamate and forms glutamine, which is our non-toxic carrier, which will then enter the bloodstream and then reach the liver, right? Okay. Now, let's say the glutamine has now reached the liver. Now what? When it reaches the liver, it's broken down to produce glutamate, and this reaction is catalyzed by glutaminase, okay? So, quick recap. We had amino acids broken down into ammonia. They combine with glutamate to form glutamine. This reaction was catalyzed by glutamine synthetase, and now we have a non-toxic carrier. This non-toxic carrier enters the bloodstream and reaches the liver, right? Where we said now it'll be converted into urea. Now, once glutamine reaches the liver, it's broken down to produce glutamate, and this reaction is catalyzed by glutaminase. Now, when you have glutamate, it's then broken down to produce alpha-ketoglutarate and ammonia. This reaction is catalyzed by glutamate dehydrogenase, right? So we had the glutamine, the non-toxic carrier, reaches the liver. It's then broken down to form glutamate, the reaction catalyzed by glutaminase. Now you have glutamate, you break it down to form alpha-ketoglutarate and ammonia, which is what we're going to be working with for the urea cycle. So the ammonia then enters the urea cycle. What about alpha-ketoglutarate? If you have the amino acid alanine and you have alpha-ketoglutarate, you can actually form alanine. Sorry, you can actually form pyruvate. The pyruvate can then be converted into glucose. This glucose is then sent to the muscles. What do the muscles do with glucose? Obviously, they'll break it down to release energy, and then we'll get pyruvate again. Pyruvate as a product can undergo a transamination reaction and form alanine, which is then sent back to the liver. And this kind of will repeat again. But our main focus right now is ammonia, and what do we do with that? Now, one thing that'll help you remember the enzyme names, because of course there are going to be a couple of enzymes in this lecture. What you can remember is that if an enzyme contains the word synthetase in its name, then that means it's going to form the substance before the word synthetase. And to give an example, you have glutamine synthetase. Now see, this enzyme has the word synthetase in it, And what is before synthetase? Glutamine. So this enzyme catalyzes the reaction that forms glutamine. This enzyme, which is carbamyl phosphate synthetase 1, we'll look at it in just in a few minutes. But see, it also has the word synthetase in it. And what does it help catalyze? It helps catalyze the reaction which forms carbamyl phosphate. Now, a second rule that you can kind of remember is that if an enzyme contains the word ase in it, then that means it's helping to break down the substance before these letters, okay, or before the word ase. Arginase, we'll look at this enzyme in a few minutes, breaks down arginine. Arginosuccinase also contains the words ase, and it helps to break down arginosuccinate. So these, like this rules will kind of help you remember the enzyme name if you kind of forget, okay? Pretty basic. Now we discussed how ammonia gets to the liver, okay? Now let's see how it gets converted into urea. So our first step is that now when we have the ammonia, we combine it with carbon dioxide and one ATP molecule, okay? And we convert it into carbamyl phosphate. This reaction is catalyzed by carbamyl phosphate synthetase 1, which we just discussed earlier. This is the reaction. You have ammonia, and sometimes what you'll see is that in certain diagrams, you'll see this bicarb ion instead of carbon dioxide. And that's totally fine because in the body, most of the carbon dioxide is in the form of bicarbonate, okay? So sometimes diagrams or like textbooks will use these two terms interchangeably, so it doesn't matter. But you have ammonia, carbon dioxide, or bicarb, plus 2 ATP molecules, okay? Now, what's important to remember here in this diagram is that this reaction is happening in the mitochondrial matrix of the hepatocyte or the liver cell. So the whole reaction is happening in the hepatocyte, correct? Or in the liver. But these two steps are happening in the mitochondria of the hepatocyte. And this is high yield to know because sometimes examiners can ask you where exactly location is this step happening. So we talked about how this reaction is catalyzed by the enzyme carbamyl phosphate synthetase 1. Now, another important thing to know here is that this enzyme has an obligate activator, okay, which is N-acetylglutamate. So this substance, which is N-acetylglutamate, won't bind to the active site, okay, because that's not where it's going to bind. It will actually bind to the allosteric site of the enzyme and activate it and cause the reaction to happen faster. So this is important to know. You also need to know that this is the rate limiting step of the urea cycle. Okay, so this is very, very important. You have to know what happens in the mitochondrial matrix. You have to know this step is catalyzed by the enzyme carbamyl phosphate synthetase 1. You have to know that this enzyme had an obligate activator, which is N-acetylglutamate. Okay, and remember that this is the rate limiting step. So you can see here that N-acetylglutamate is activating this enzyme. Now, we combined ammonia with carbon dioxide to produce carbamyl phosphate, and the reaction was catalyzed by carbamyl phosphate synthetase 1. Now let's come to the second step where we combine carbamyl phosphate with ornithine, and what do we get? We get citrulline. This reaction is catalyzed by ornithine transcarbamylase. Okay? Again, you can see that this reaction is in a gray box, and what does that indicate? It indicates this is happening in the mitochondrial matrix. Okay, now the rupture of a high energy phosphoanhydride bond facilitates the carbamyl group from being transferred to ornithine and then forming citrulline. Okay, and remember this reaction is catalyzed by ornithine transcarbamylase. Now you can see the rest of the steps of the urea cycle happen in the cytoplasm. So once you have the formation of citrulline, it then moves on to go to the cytoplasm. Now, the third step is the formation of arginosuccinate. We had the citrulline from step 2, and we combine it with aspartate to form arginosuccinate, okay? Now, what's important to remember here is that during this reaction, one ATP molecule is hydrolyzed twice, okay? So you take an ATP molecule, combine it with water, and you get AMP and this substance, which is called pyrophosphate. The pyrophosphate is then broken down to give you two inorganic phosphates, and this reaction is catalyzed by the enzyme pyrophosphatase. If you remember the enzyme rules that we talked about earlier on in the lecture, I mentioned that if something has the word ase in it, it kind of indicates that whatever comes before that is what is being broken down. So pyrophosphatase, you can kind of give you a hint that you're going to get inorganic phosphate from this enzyme. Okay, so pyrophosphate is then broken down to give you two inorganic phosphate. Now, the reaction where we combine citrulline and aspartate to give us arginosuccinate is catalyzed by arginosuccinate synthetase. Again, we use the enzyme rules that we talked about earlier on. What is being synthesized here? Arginosuccinate, because the name tells you. It has the word synthetase in it. Now, allows the transfer of one nitrogen atom into the urea molecule from aspartate. So aspartate gives us one nitrogen atom to form the urea molecule. So we see here we have citrulline from our step 2, combined it with aspartate, catalyzed by arginosuccinate synthetase, and gives us what? Arginosuccinate, because this enzyme name tells us that. And one ADP molecule is hydrolyzed twice. So here we see pyrophosphate, which is broken down to give us two inorganic phosphate, this reaction being catalyzed by pyrophosphatase. This is our third step. Citrulline, okay, from step 2, combining with aspartate, one ATP molecule, and giving us arginosuccinate, this reaction being catalyzed by arginosuccinate synthetase. Coming to step 4, where we have formation of arginine and fumarate. Now, what happens here is the enzyme arginosuccinase cleaves arginosuccinate, which we had from step 3, into arginine and fumarate. Now, the enzyme name again gives you a hint as to what it does. This enzyme catalyzes a reaction where arginosuccinate breaks down into arginine and fumarate. It contains the word ase in it. And what is it breaking down? Arginosuccinate. Okay? You're getting fumarate and you're getting arginine. Fumarate actually goes on to be used in the Krebs cycle. Okay? Arginine is actually further broken down and cleaved. And we'll see that. So we have arginosuccinate, broke it down into arginine and fumarate. We said fumarate is used in the Krebs cycle. Arginine we're going to talk about now. So step 5 is the formation of ornithine and urea. So where do ornithine and urea come from? Okay, they come from here. Arginine is cleaved by the arginase enzyme into ornithine and urea. Okay, now again, the enzyme that we have here is arginase. This again contains the word ase in it. And so what is it breaking down? Arginine. Arginine is cleaved to give us ornithine and urea. Ornithine, of course, goes back, okay, to the first step and picks up another carbamyl group from the carbamyl phosphate. And now we have our end product, okay, which is the non-toxic form, which is urea. Urea is then transported to the kidneys to be eliminated from the body. So essentially what we did is that when a person eats proteins, they're broken down to amino acids. Amino acids, okay, are broken down to form ammonia. What's the problem? Ammonia, if accumulates inside your body, is toxic, okay, so we have to get rid of it. How do we do that? We've converted into urea. The first step we talked about is that because ammonia is toxic, we have to combine it with a carrier. We combine it with glutamate to form glutamine. The glutamine then entered the liver, then entered the bloodstream, and then went to the liver. And then, once it reached the liver, we broke it down again to form glutamate, and the glutamate formed alpha-ketoglutarate and ammonia. And then we took that ammonia and put it into the urea cycle, which we just talked about. Okay? Here. This is, now we get the ammonia, right? And then we put it into the urea cycle, which is everything that we just talked about in the last few minutes. Let's just go over it one more time. We got ammonia, we combined it with carbon dioxide and 2 ATP molecules and converted it into carbamyl phosphate. Carbamyl phosphate synthetase 1 catalyzes this reaction, and here, it's not shown here, but you have an obligate activator of this enzyme, which is N-acetyl-glutamate. Then, for the second step, we took carbamyl phosphate, and then we combined it with ornithine and converted it into citrulline. This reaction was catalyzed by ornithine transcarbamylase. Also, it's important to keep in mind that these two reactions are happening in the mitochondria of the hepatocyte or the liver cell, okay? It's high yield. Then citrulline combines with aspartate and you have an ATP molecule that's being used here. It's important to keep in mind that this ATP molecule is being hydrolyzed twice. So ATP is converted into AMP plus a pyrophosphate. Pyrophosphate is broken down to give you 2 inorganic phosphates. Then citrulline plus aspartate gives you arginosuccinate, and this reaction is catalyzed by arginosuccinate synthetase. Remember the enzyme rules that we talked about. Arginosuccinate is then converted into arginine and fumarate. This reaction is catalyzed by arginosuccinase. Fumarate is actually will be utilized in the Krebs cycle. Arginine is taken and further broken down to urea, which is what we were trying to get, right? Trying to convert the ammonia into. And we also get ornithine. This reaction is catalyzed by the enzyme arginase. Remember to keep those enzyme rules in mind. The ornithine then cycles back, okay, to pick up another carbamyl group from a carbamyl phosphate, okay? Now, this is like an overview of the urea cycle. These are 2 equations that you will sometimes see, okay? Now, overall, 3 ATP molecules are used in the urea cycle, but 4 high energy phosphate bonds are broken to release energy. Now, we already talked about why this is. 3 ATP molecules are being used, but you have 4 high energy bonds being broken. So just to quickly recap as to why that is, you have ATP converted into ADP, okay? There are 2 reactions, and these are 2 high energy phosphate bonds. This is the 3rd high energy phosphate bond where you have ATP being converted into AMP and giving you pyrophosphate. The 4 high energy or the 4th high energy phosphate bond being broken is when pyrophosphate is being converted into 2 inorganic phosphates, okay? So this is just to kind of explain this statement to you. Now, when you're trying to form the urea molecule, okay, 1 carbon atom comes from carbon dioxide, okay, the first step that we talked about. 1 nitrogen comes from the ammonia, okay, and 1 nitrogen atom comes from the aspartate. We discussed all of this when we were talking about each of the steps. It's also important to remember that ornithine acts as a carrier of various atoms in the process of synthesizing urea, okay? So we talked about how when you get ornithine from arginine, it just cycles back to pick up another carbamyl group from carbamyl phosphate, okay? So it acts as a carrier. This is again just to emphasize the locations of each of the steps in the urea cycle. That formation of carbamyl phosphate and formation of citrulline happen in the mitochondrial matrix of the hepatocyte or liver cell. And that the formation of arginosuccinate, cleavage of arginosuccinate into arginine and fumarate, and the hydrolysis of arginine into ornithine and urea all happen in the cytoplasm of the hepatocyte or the liver cell. Now, if you remember, we talked about the formation of fumarate, correct? And we said that fumarate will be used in the Krebs cycle. So fumarate actually, technically speaking, is what kind of connects these 2 cycles together. The citric acid cycle or the Krebs cycle and the urea cycle are closely related. In the urea cycle, one of the byproducts that we discussed was fumarate. Fumarate can be converted into malate, which can then be converted into oxaloacetate. Oxaloacetate can be converted into aspartate, which is what gives us the 1 nitrogen atom to form urea. So oxaloacetate, of course, can be utilized in the Krebs cycle itself, but it can also be converted into aspartate, which we said is what gives us one of the nitrogen atoms to form urea. Just give me one moment. All right. So we've discussed the whole urea cycle. We got an overview of it, and we also talked about how the urea cycle and the Krebs cycle are closely related, okay? Now let's discuss the regulation of the urea cycle. The urea cycle is regulated by mainly two things: the enzyme levels and the concentration of the cycle intermediates, right? Both of these are extremely important things. If you don't have the enzymes, of course, you can't convert any of the products to other products in the cycle. Also, if there is high or low intermediates concentration, then that kind of dictates how often or how frequently the urea cycle will happen. Okay? Now, certain reactions directly produce cycle intermediates. For example, if someone takes arginine from the diet, it can be converted directly into ornithine, which we talked about is utilizing the second step of the urea cycle. Glutamate can be converted into ornithine by intestinal enzymes as well. Now, the fine regulation of the urea cycle occurs through the regulation of carbamyl phosphate synthetase 1. Okay? Now, if you recap, we mentioned that this enzyme, okay, is catalyzing the rate limiting step, okay, which is why it's really important. So that's why fine regulation of the urea cycle happens when you regulate this enzyme. You also remember we talked about N-acetyl glutamate, which we said was an obligate activator or heterotropic allosteric activator, and I mentioned how it binds to the allosteric site of the enzyme, which in this case is carbamyl phosphate synthetase 1. So, N-acetyl glutamate is synthesized in the liver from acetyl-CoA and glutamate, okay? This reaction where we get N-acetyl glutamate is catalyzed by N-acetyl glutamate synthetase. Again, those enzyme rules are really important because this name has the word synthetase in it, and what is it synthesizing? N-acetyl glutamate. So the concentration of N-acetyl glutamate is determined by two factors, okay? The concentration of the substrates, okay, which are acetyl-CoA and glutamate, we need these to make N-acetyl glutamate, and the concentration of arginine, which activates the N-acetyl glutamate synthetase, or the enzyme which is required to catalyze the reaction of the formation of N-acetyl glutamate, okay? Which makes sense. You need to have the substrates, you need to have the enzyme to form N-acetyl glutamate. So remember that urea synthesis is a cyclic process, okay? And while ammonium ion, carbon dioxide, ATP molecules, and aspartate are all being consumed in this process, you're not gaining and you're not losing ornithine, citrulline, arginosuccinate, or arginine, okay? Because remember, this is a cycle, okay? You don't gain these products, you don't lose any of these products, okay? Or any of these substances. But you do consume ammonium ions, carbon dioxide, ATP, and aspartate, okay? Now, why do we actually have to know this whole cycle and all of enzymes, okay? Why is it important? Being that sometimes in some children, okay, or some infants, there is a genetic deficiency in some of these enzymes, okay? And if these enzymes are not functioning properly, then that can lead to certain diseases or certain problems, because we talked about that ammonia is toxic, okay? And we have to get rid of it. So, urea cycle disorders are inborn errors of ammonia, particularly ammonia, the enzymes that are supposed to break down ammonia, okay? Now, disorders of the urea cycle can result from defects in any of the following 6 enzymes, okay? If any of these enzymes are not functioning properly, and also N-acetyl glutamate synthetase, because this is required to form what? N-acetyl glutamate, which is what? The obligate activator of carbamyl phosphate synthetase 1, okay? Which is why we're also including it here. In the slides, it's mentioned arginosuccinic acid lyase, which is the same as arginosuccinase, okay? So you should expect a question from a deficiency of carbamyl phosphate synthetase 1 because this is the rate limiting steps enzyme, okay? And also ornithine transcarbamylase because this is the most common disorders of urea deficiency, okay? If there is a defect in urea cycle, what would happen? You would have hyperammonemia, which is excess ammonia or ammonia in the body, which makes sense, right? Because you can't convert this ammonia into something else because the enzymes aren't functioning properly. You're also going to have elevated blood glutamine, which again is also pretty self-explanatory because what? We combine the ammonia with glutamate to give us a non-toxic carrier, right? But because we can't use the ammonia, we can't utilize it, we're going to keep converting it into a non-toxic carrier, but can't do anything with it after that. So that's why you'll have an elevated glutamine and also an excess of ammonia. You're also going to have a decreased blood urea nitrogen. So you're going to have a decrease in urea because, again, the enzymes aren't functioning, so you can't form urea in the first place. So during the 24 to 72-hour postnatal period, symptoms of lethargy, vomiting, and hyperventilation can happen. And if you don't do something about this, it can end up resulting in coma or death. Okay, so this is pretty serious. So what happens? Let's discuss starting off with what if carbamyl phosphate synthetase 1 is deficient. Okay, you're going to have hyperammonemia, you're going to have an excess ammonia in the body because, of course, you can't utilize it in the urea cycle. The first enzyme isn't functioning properly. If this step doesn't happen, none of these steps can happen. Okay, you're going to have an increase in glutamine. Why? Makes sense. You have ammonia combined with glutamate formed a non-toxic carrier, but nothing after that because the first step of the process isn't working or isn't functioning properly. This point actually is important. No orotic acid formation. Okay, why is this important? Because this point actually helps you differentiate in a question or in a case scenario if this child or this infant has a carbamyl phosphate synthetase deficiency or if they have ornithine transcarbamylase deficiency. Okay, because you'll see here that if a child or infant has ornithine transcarbamylase deficiency, they're going to have an excess of orotic acid formation, okay? They're also going to have hyperammonemia, just like here, but here they also have orotic acid and uracil formation. And the excess of orotic acid actually accumulates and results in orotic aciduria, okay? Orotic acid and uracil are actually pyrimidine synthesis intermediates. And what I looked up is because when you get the formation of carbamyl phosphate, but this enzyme isn't working, this is actually shunted to the pyrimidine synthesis pathway, okay, and gets converted into orotic acid, which is why if an infant has this enzyme's deficiency, carbamyl phosphate gets converted into orotic acid, and they will have an excess of orotic acid or orotic aciduria. Okay? Again, glutamine, glycine, alanine, this point is the same in both. But the main differentiating factor between these two deficiencies is that there is an excess amount of orotic acid in deficiency of ornithine transcarbamylase. Okay? So we differentiate between OTC and CPS deficiency based on the presence of orotic acid in the urine. Now, ammonia and glutamine, okay, if there are excessive amounts, they can go to the brain and accumulate and can cause brain damage because both are toxic, but the patient will have severe neurological abnormalities, okay? Glutamine in very high amounts, and if it ends up reaching the brain, then it will become toxic and cause neurological abnormalities in the patient, okay? Glycine and glutamine will accumulate, build up, okay? And alanine is another amino acid that accumulates due to hyperammonemia due to urea cycle defect, okay? So just remember these points. It's pretty self-explanatory. Because ammonia is toxic, it will cause neurological abnormalities in the patient, okay? So what do we do about this? Now, we talked about how proteins are broken down to amino acids and the amino acids catabolize to give us ammonia. So what can we do in this case? We can give the patient a low protein diet because low protein means less amino acids, which means the urea cycle won't happen frequently. Okay, because of the excessive glycine, okay, we can give the patient benzoate to remove this glycine as hippurate, and we can give the patient phenylacetate to remove the glutamate as phenylacetylglutamine, okay? Because we talked about how these are also increased amounts inside the body, so we can feed the patients these substances to remove them. Now, arginosuccinase enzyme deficiency, which is this enzyme, which converts arginosuccinate into arginine and fumarate. Again, treatment here is to give a low protein diet, okay? But you also want to give a diet that's high in arginine, okay? And why is that? Why would you want to give a diet that's high in arginine? Because arginine, if you remember, I mentioned that arginine activates the enzyme N-acetylglutamate synthetase, which is the enzyme required to form N-acetylglutamate, which is an obligate activator of carbamyl phosphate synthetase 1. Okay? That's why you want to give an enzyme, you want to give arginine, okay, in the deficiency of arginosuccinate, in this deficiency, arginosuccinate is secreted instead of urea. So you still want to give a low protein diet because you don't want an excessive amount of ammonia in the patient. But here, because you reach till this point in the urea cycle, since these two enzymes are working and this one as well, this will now be excreted from the body, okay, in the place of urea. Like we discussed before, hyperammonemia can lead to excess glutamine, okay? Alpha-ketoglutarate plus ammonia, or ammonium, will give us glutamate plus ammonium and give us glutamine. We said before as well that high glutamine and glutamate may lead to brain damage, possibly by producing osmotic effects that cause brain swelling. Okay, just to give you like a bit of detail, it's not really important to remember, but you just have to know that they are harmful if they accumulate in the body in high amounts. But they can kind of pull water, okay, because of their high osmolality, they can pull water and cause brain swelling. Okay, they're very strong osmoles. So if they're present in the plasma, or if they're present in any solution, they can make the solution very concentrated. Since the body compensates by pulling in more water to kind of stabilize the osmolality of a solution. That's like a side detail. It's up to you just to kind of make you understand what we're talking about. So if you're in the exam, how do you know that this question is talking about urea cycle or urea cycle disorders? Usually you see a neonate and you see hyperammonemia. You will see excess glutamate or glutamine, and you will see excess alanine and encephalopathy, seizures, and coma. Because we talked about that if you don't treat the symptoms earlier on, it can lead to coma and other neurological abnormalities because of the toxic effects of ammonia. So a few questions that we can do for this or for this session are these ones. So most of the ammonia is carried in the circulation as what? Okay. So this point we mentioned earlier on that because ammonia is toxic, we can't allow it to go into the bloodstream. Okay. And there we mentioned that we combine it with a substance. And what is that substance? Exactly. It's basically combined with glutamate and it gives us glutamine. So ammonia combined with glutamate gives us glutamine, which is the non-toxic carrier form of ammonia. This is like basically a simple recall question. Now, which one of the following enzymes is catalyzes the regulated step of the urea cycle? Again, pretty simple active recall question. Okay. The enzyme that catalyzes the rate limiting step is carbamyl phosphate synthetase 1. So an individual harboring a mutation in ornithine transcarbamylase, this should be carbamylase, apologies, would be expected to exhibit which of the following measurable abnormalities? So here, the answer is elevation in blood orotic acid. And if you remember, we talked about this point being the differentiating factor, okay, that kind of tells you this infant or this child has ornithine transcarbamylase deficiency. Because when this enzyme doesn't work, you have an accumulation of carbamyl phosphate. And then it gets shunted to form pyrimidine. And borotic acid is an intermediate in the pyrimidine synthase pathway. Now, a 2-day-old infant is brought to the ER suffering a seizure. Analysis of serum ammonium levels revealed to be quite high. Suspecting a urea cycle impairment, the ER physician initiates hemodialysis, which reduces the ammonium levels. After the hemodialysis, the infant is treated with supplemental arginine, which maintains the reduction in serum ammonium. The ability of arginine to render this effect stems from its role in activating the synthesis of an activator of one of the enzymes of the urea cycle. Which of the following represents this potent activator whose synthesis is induced by arginine? This is again a pretty basic active recall question. The answer to this would be N-acetyl-glutamate. Okay, that we talked about how arginine actually activates the enzyme that forms an acetyl-glutamate. Okay, so yeah. Now, a newborn that developed seizures 2 days after birth was diagnosed with elevated serum ammonium, glutamate, and alanine. A neonatal urea cycle defect was suspected, so a urinalysis was performed to assay for orotic acid and serum assayed for citrulline. The levels of both were found not to be elevated, a defect in which of the urea cycle would most likely account for the symptoms observed in this infant. Okay. Here, what's important to know or to note in this question is that they tell you the levels of both were found not to be elevated. Both here in question are citrulline and orotic acid. What does that tell you? Since you have no orotic acid being formed, that means there is no carbonyl phosphate being formed because carbonyl phosphate is shunted into the pyrimidine synthesis pathway, and that's how we get orotic acid. If both levels are low, that means the deficiency is in the carbonyl phosphate synthetase 1 enzyme. Okay? Because you don't have orotic acid, you don't have citrulline. This means they aren't being formed. So before this enzyme, this is this enzyme, which is carbonyl phosphate synthetase 1, which is deficient in this infant. Now, a deficiency of arginosuccinate synthetase can be suspected in a neonate with elevated serum ammonia. To distinguish the fact that hyperammonemia is indeed due to this enzyme and not due to a deficiency in any other urea cycle enzymes, one can assay for the serum concentration of citrulline and orotic acid. Which of the following would best describe the expected findings? Okay. Here, what you need to note is that they're telling you a deficiency of arginosuccinate synthetase. This is what you're trying to see, okay? In a situation where this enzyme is deficient, they're asking you what would you see in an infant or a neonate. So citrulline levels will be 300 times normal, and orate will be near normal, or orotic acid, okay? Why is that? The mutation lies in arginosuccinate synthetase, okay, which is this enzyme, which means citrulline can't combine with aspartate to give us arginosuccinate, okay? So that's why citrulline is being accumulated, okay? And that's why citrulline levels are very high. But ornithine transcarbamylase levels are normal, okay? Which means that ornithine and carbonyl phosphate are being combined to give us citrulline. Carbonyl phosphate is not accumulating inside this person, which means it's not being shunted to the pyrimidine synthesis pathway, which means it's not giving us orotic acid. That's why orotic acid levels will be near normal, correct? But citrulline levels will be very high because this enzyme is the one that's not functioning. Now, a child was born at home and developed seizures 2 days after birth. Upon examination in the ER, it was found the infant had elevated serum ammonium, glutamate, and alanine. The ER physician suspected that the child was suffering from a defect in the urea cycle and ordered a urinalysis to assess the levels of orotic acid and the serum assay for the level of citrulline. Orotic acid levels were above normal and citrulline was barely detectable, a defect in which enzyme of the urea cycle would most likely account for the symptoms observed in this patient. Okay. Again, the important part here is orotic acid levels were above normal and citrulline was barely detectable. So what does this tell you? Citrulline is not being formed, which is why it's barely detectable. Orotic acid levels, very high, which tells you what? Carbonyl phosphate is being formed, okay? So it's not a deficiency of carbonyl phosphate synthetase 1, right? And the carbonyl phosphate is actually accumulating and converting into orotic acid. But orotic acid, sorry, not orotic acid, carbonyl phosphate is not being converted into citrulline. So which enzyme is exactly deficient? It's ornithine transcarbamylase. And that's it. But guys, remember that any question that you see about urea cycle, okay, or urea cycle disorders, you will have an infant, okay, because there are what? There are genetic abnormalities of the enzyme. So you'll see symptoms very early on, okay? If it's not a child or not an infant, it's very, very less likely for it to be an ammonia disorder because you're going to see the symptoms very early on. Another thing that you should take care of is that which intermediate levels are they telling you are high and which levels are low. Like here, they said orotic acid levels are high and citrulline was barely noticeable, okay? But in the previous question, we said citrulline levels were very high, okay? And we were talking about arginosuccinate synthetase deficiency. So these things are what kind of give you a hint as to which deficiency are you dealing with or what mutation you're dealing with. I think that the questions from this lecture shouldn't be very difficult, okay? The concept itself is simple. So inshallah, they'll give you easy questions from this. And that's it. If you guys have any questions, my email and phone number are here. You can text, you can email, whatever you guys prefer. And inshallah, you guys got this. Also, please scan the QR code to give us feedback about this session. And good luck.